This is Andrew Bolte. In the first 18 months of his life, Andrew was developing as any other child would, starting to speak his first words. At a routine checkup with his doctor, fluid was found in Andrew's ear. After examination, the physician diagnosed him with an ear infection. Naturally, the doctor immediately prescribed antibiotics for Andrew to kill the pathogen. Andrew went through one course of antibiotics, but the infection persisted. The doctor prescribed another course, and there was again no change. Andrew endured 6 courses of antibiotics and still had fluid in his ears. The doctor then came to the conclusion that the fluid was not due to infection, but the result of a milk intolerance. Once milk was removed from Andrew's diet, the fluid cleared up, but it was too late. Andrew began exhibiting abnormal behaviors. He would not make eye contact, he had trouble walking, and refused to eat solid food, instead eating things like paper and dried paint from the wall. Unsurprisingly, Andrew also had severe gastrointestinal symptoms. This is Ellen Bolte, Andrew's mother. After seeing the decline in her son's behavior and cognitive function, she took Andrew to see several so-called experts. Ellen refused to believe there was no hope for Andrew. She had no medical or scientific background, but began researching possible causes, searching for answers. She ended up reading a paper proposing a link between intestinal overgrowth of the Clostridium bacteria, which is known to survive the attack of most commonly prescribed antibiotics, and autism. Of course, Ellen immediately thought that this could be the answer she was looking for. She also found that there was one antibiotic, vancomycin, that could specifically target Clostridium. Eager to help her son and with no other options, Ellen began writing to doctors to prescribe Andrew vancomycin. Most of the doctors wanted nothing to do with a risky experiment like this, and the others referred her to other specialists. Over the next 15 months, Ellen had reached out to 37 doctors until she finally found a gastroenterologist who decided to test her hypothesis. Within weeks, Andrew's cognition and behavior saw dramatic improvement. He got potty trained, started to speak again, and was even putting together puzzles. Ellen had new hope for her son. Unfortunately, once the 8-week course was completed, Andrew regressed again, displaying much of the old dysfunction that he had dealt with for most of his life. The powers that be and those controlling the media and the healthcare system not only ruined Andrew, but also gave up on him. To think a doctor didn't even hesitate to give Andrew that quantity of antibiotics without thinking of other possible causes or potential issues with that treatment is honestly sickening. Then for the specialist to basically tell Ellen that it just happened and that there was nothing she could do to help him just speaks to the negligence of the system. It took 36 doctors who refused to take a chance and think outside the box to finally find one who was open to trying it. This is our society. Many people just accept that the root cause of autism is purely genetic. While the research indicates that genetics increase vulnerability to developing autism, some people with these genetics will not be affected. In the case of Andrew, the problem was clearly not a genetic one. He only developed the condition after the antibiotics. So what is the cause of autism? It's important to note that there may not be just one cause, and there are no hard endpoints with autism diagnosis. It's not a disease like diabetes where we can measure blood sugar and use that as diagnosis. However, there has been tons of research done on environmental causes that are likely factors. The first place we need to investigate is the intestinal microbiome, an incredibly complex array of thousands of types of bacteria living in our guts. There's so much we don't know about the microbiome, but there's also a considerable amount that we do know, especially when it comes to autism. One thing that's been consistently found is the significantly higher levels of Clostridia bacteria in the guts of autistic children in comparison to healthy children. How could this have occurred? Like we saw with Andrew, overuse of antibiotics could definitely cause this imbalance. The problem is that most conventionally used antibiotics are not effective in killing Clostridium, but are very effective at wiping out many other types of bacteria. This gives Clostridium less competition for nutrients and more space, the perfect conditions for it to spread like wildfire. Other children are diagnosed with autism very early on and without any exposure to antibiotics, but that doesn't necessarily mean that they can't have this problem. Babies in the womb are essentially free of any bacteria, but the vaginal fluid acts as a microbiome starter pack for the child. When the child is passed through the birth canal, they will inherit these microbes from the mother. The mother's milk also has its own microbiome, which is how the children's own gut microbiome begins to develop. Without contact to the vaginal fluid during birth or access to the custom-made bacteria of the mother during breastfeeding, children become much more prone to having disrupted gut biomes. Children who are delivered by C-section and children who are bottle-fed both have higher rates of autism than those delivered vaginally and breastfed. You may be thinking, how could a problem with bacteria in the gut possibly affect the brain? Clostridium are capable of degrading the protective mucus layer within the intestines, making the intestinal barrier susceptible to damage and eventually permeability. The intestine is normally selectively permeable, allowing in nutrients from the food we eat. But if it becomes too permeable, it allows unwanted components into the bloodstream. Once the barrier has become compromised, the rest of the body becomes exposed to the potential neurotoxins that Clostridia can produce. These toxins can block the release of important neurotransmitters or increase the release of others, throwing off the balance of these important behavioral regulators in the brain. Some toxins produced by certain Clostridium bacteria can even induce a process known as necrosis. I've explained in other videos that apoptosis is the way that cells normally die, in a controlled manner and intentionally done so by the body. If you want to imagine apoptosis as someone passing away in their sleep and being gently laid to rest in a grave, then necrosis would be like if someone got stabbed several times and left on the street. It is an uncontrolled cellular death that the body does not want. Another worrisome metabolite that Clostridium can secrete is propionic acid, or PPA. Now, short-chain fatty acids like PPA are normal to a certain extent in the gut, but in high concentrations they can have adverse effects on brain function. When PPA is injected into rats, these rats go through severe behavioral changes, replicating those of autism.
Dr. McFabe's control subjects exhibit typical behavior. Rats are highly social animals. They greet by sniffing nose to tail and interact as they explore their surroundings. But when a propionic acid solution is introduced, this all changes.
What we noticed remarkably within 2 minutes of putting this propionic acid in, this animal immediately starts exhibiting some very peculiar behaviors. Obviously, he's hyperactive and moving very quickly, and then repetitive behaviors. Here you see an abnormal posture of walking with a hunchback. And it's an unappreciated part of autism in that patients often have toe walking, abnormal posturing, arched back. So now we'll place another animal that's been given propionic acid. And a peculiar thing happens. They show none of the normal social greetings of two rodents seeing each other.
A video tracking system records and compares the obsessive and antisocial PPA-treated rats with the normal rats. Other experiments showed responses such as rigidity and object fixation, and the intriguing fact that the effects of PPA wear off.
So now we're about a half an hour after we've exposed these two animals to propionic acids, and their behavior afterwards reverts back to normal, and it's a fat, so it's burned through metabolism. The animals become more social. Usually after their escapades, they tend to just go back to sleep. So again, we have a reversibility of hyperactivity and a reversibility of social behavior.
PPA is also sometimes used as a food preservative and is commonly produced by carbohydrate fermentation. This could explain why autistic children tend to crave refined grains. If it sounds a bit ridiculous that microscopic bacteria could control the cravings of a human being for its own survival, consider this nasty bug. Toxoplasma parasites can infect mice, but this isn't their ideal camping grounds. They'd much rather be in the gut of a larger animal that has more space and resources. So, when they're present in a mouse, they cause the mouse to act without any sense of fear, walking out in broad daylight and even confronting cats. These are great strategies to ensure the mouse gets eaten by a predator and Toxoplasma's mission is complete. So yeah, microbes can definitely have a profound effect on cognitive behavior. Other than antibiotics, two environmental toxins that people are unfortunately far too exposed to nowadays have been strongly correlated with autism. These are acetophetamine, found in painkillers like Tylenol, and glyphosate, the highly controversial herbicide used on crops. While these may seem like they have nothing in common, the way that they could negatively impact cognition is strikingly similar. When ingested, acetophetamine undergoes xenobiotic metabolism. Basically, the body perceives it as a poison. The body uses sulfate and glutathione in order to detoxify it, so the overuse of these drugs can deplete these molecules. Glyphosate has the ability to inhibit gut bacteria that synthesize methionine, an amino acid that serves as a precursor for both sulfate and glutathione. Sulfate is particularly relevant here because it participates in the enzymatic regulation of several neurotransmitters. Perhaps non-coincidentally, autistic children have lower plasma levels of sulfate and lower activity in enzymes that require sulfate. Since methionine is also needed for glutathione synthesis, glyphosate can also deplete glutathione, the body's master antioxidant. This will result in increases in oxidative stress, which will in turn inhibit important enzymes that regulate the body's system of methylation. Elevated markers of oxidative stress and depletions in glutathione are commonly seen in autistic patients. Methionine is important for generating sulfate and glutathione, but it is also crucial for methylation, which is critical to mental health. Without getting too deep into it, methylation is the process of altering the activity and function of various biological compounds. Quite a few molecules that are dependent on methylation have huge effects on mental health, such as glycine, creatine, and dopamine. In particular, inadequate methylation of dopamine could cause mental states that would be considered inflexible, as Chris Masterjohn explains in his talk on methylation.
If you don't have enough methylation, and you have too much tonic dopamine, too much background noise, too much C level, you will not perceive the phasic dopamine pulse as having as meaningful as a rise above the background noise. And you will therefore have a pathological level of inflexibility. You'll have too much mental stability. In other words, you'll be mentally unflexible, and this can form brittle mental states.
This experiment studied people's emotional response to looking at unpleasant pictures using brain scans. Those with low methylation had a much greater emotional response to the pictures and also had a harder time getting the pictures out of their head. These aren't necessarily autistic behaviors, but there are certainly similarities between this brittle mental state and the way we see autistic children process information. Additionally, methylation is an important part of regulating our DNA. What's important to know is that DNA is just a blueprint, a master library of instruction manuals for building different proteins. Alterations to the DNA, such as methylation and demethylation, affect the likelihood that a certain protein gets synthesized. Proteins are fundamentally necessary to create normal functioning body tissue, so healthy methylation will ensure that we're getting all of the proteins made that we need and in the right amounts. However, it has been shown that those that suffer with autism spectrum disorders have abnormal DNA methylation patterns. As we discussed, toxins like painkillers and herbicides are harmful to this crucially essential system in the body by depleting methionine. However, we don't need to synthesize methionine. It is abundant in dietary protein, especially animal protein. This brings me to my next point, how nutrition can have important implications for autism spectrum disorders. Nutrients like vitamin B12, folate, magnesium, and choline all support the system of methylation, but neurological function goes so much further than just methylation. We also need to consider all of the nutrients necessary for building proper tissue, those necessary for energy metabolism, and all of the other systems in the body. In other words, we need to emphasize nutrient-dense and complete foods. The issue is that this is incredibly difficult since children with autism spectrum disorders often reject whole foods in favor of processed snacks. Chris Kresser describes a child he worked with who was on the autism spectrum and who had seen several doctors beforehand with little to offer outside of prescriptions. Kresser suggested a change in diet, and the effect was incredible.
So we started treating him, which wasn't easy because of his OCD-like tendencies. It was really hard to get him to change his diet. So we just started focusing just on those mechanisms that I talked about, getting him some more nutrients and treating his gut and dealing, trying to get some of those things out of his food. Initially, just switching those brands of toaster waffles so that at least they were made from stuff that he wasn't clearly reacting to, and then gradually over time shifting. Within several weeks, he was having fewer tantrums. He was less set off by the things that would have done that before. About 4 months into the treatment, his teacher called home from school and was like, "Where's Leo? What have you done with Leo? And who's this guy you're sending to school in his place?" Because it had been horrific for her as his teacher at school, and to the point where his parents often had to come pick him up and bring him home from school because he was so disruptive. Then I talked to Leo's mom maybe 6-7 months into the treatment. By then we had the follow-up test back. A lot of the issues that we set out to address had been resolved. His diet had expanded significantly. He was eating foods that he would have thrown against the wall just a few months before. He was more tolerant, more affectionate, less controlling and rigid, and just better adjusted kid overall. And at the end of that time, his mom said something that really struck me, and it's why I wrote the book. She said, "Why don't more doctors know about this? Like, there's so many kids out there like Leo who are suffering from these kind of behavioral disorders, but neither their parents nor their doctors are even looking at this other stuff like the diet and the gut and all of these things. So it's not even a consideration."
While there are several nutrients the body needs to obtain from diet in order to maintain good mental health, it's just as important to focus on eliminating compounds in food that could be detrimental to cognition. Let's take a look at BCM7 and GM7. These are peptides that are a result of casein, a protein found in most dairy, and gluten digestion, respectively. These metabolites can act on mu-opioid receptors in the brain, having some striking effects. First off, too much stimulation of the mu-opioid receptor can cause the receptor to become downregulated, meaning the body has a reduced sensitivity to the compound. This is a hallmark of drug addiction, of course, and why opioid addicts will need to keep upping their dose to feel the same effect. This is significant in autism because these mu-opioid receptors are key in our ability to learn. This study genetically altered mice to have no mu-opioid receptors and compared them to normal mice in their ability to navigate through a maze. As you can see, mice lacking the mu-opioid receptor struggled mightily in learning how to get through the maze over time. Another issue with these compounds is their effect on genetics. As we discussed, DNA methylation is vital to healthy neurological functioning. BCM7 and GM7 derived from dairy and gluten inhibit the uptake of cysteine, leading to decreased levels of glutathione and SAM, a key substrate involved in methylation.
Gotta get the bread and milk. I gotta get the bread and milk.
As you can see, autistic children have also been shown to have reduced levels of glutathione and SAM. So would a gluten-free and casein-free diet help with autistic symptoms? While there are anecdotes of children improving on these elimination diets, the body of evidence is less than convincing. It's entirely possible that the damage has already been done, and reversing it could take multiple years on the diet to show any true promise. Another food compound that could contribute to autism is oxalate. With the recent popularity of the carnivore diet, many people are becoming aware of the toxic effects of this molecule, found in a variety of plant foods. Oxalate can form calcium crystals that can lodge themselves into different parts of the body, impairing function of those tissues. Here's a look at an oxalate crystal forming within the axon of a nerve implicated in neuropathy. Oxalate is a neurotoxin for this reason, and it can have a huge effect on cognitive function. One remarkable study looks at the horrifying effects of oxalate toxicity. Starfruit has been well established to be a neurotoxin, so researchers began to investigate what exactly in the fruit was giving it this property. They fed rats who had one of their kidneys removed one of a few foods, either starfruit juice, or pure oxalate. They removed one of their kidneys so that they would have an impaired ability to filter out toxins. All of the mice who had the starfruit juice, which is devoid of oxalate, were fine. However, every single one of the mice who ate either starfruit or oxalate showed severe movement and cognitive dysfunction. Even scarier is that 2 of the mice in the starfruit group and 4 of the mice in the oxalate group died within minutes after they were seen exhibiting these behaviors. The results were clear as day. The oxalate was the source of the toxicity of starfruit. You might be thinking, well, the mice didn't have one of their kidneys. If they did, this probably wouldn't be as big of an issue. Well, that would be true, except oxalates are known to impair kidney function. In fact, 80% of all kidney stones are oxalate crystals. Have you ever wondered why antifreeze is so toxic? When it gets metabolized by the body, it gets partially converted into oxalate.
We're going to put antifreeze on bologna. What? Why, Papa? Why? Because it eats the intestines. That's why.
I recommend watching Sally Norton's talk on oxalates to see just how bad high levels of oxalates are. One risk for having high levels of oxalate is, you guessed it, a gut microbiome imbalance. Certain microbes in the gut can safely metabolize oxalate, but when you consistently nuke them with antibiotics and other toxins, your body has little defense against them. Autistic children have incredibly high levels of oxalate in the urine when compared to controls. Additionally, there seems to be much more anecdotal evidence for the efficacy of a low oxalate diet in autism than there is for a low casein or low gluten diet. So we've covered a lot of different potential factors that could contribute to autism. While they may all seem like they're separate issues, there's a lot of overlap. The research continues to show that it is not our genetics, but our modern lifestyles that are the main reason autism is on the rise. Gut microbiome imbalances from the overuse of antibiotics or exposure to oxalate and glyphosate appear to have some role in autism. Casein and gluten are more abundant in food than ever and may also have a role. Having a child by C-section and feeding them formula from bottle is common nowadays, but could also have an impact on these disorders. Parents don't think twice about giving their kids Tylenol for a headache. This video just scratches the surface of potential causes, but one thing that is abundantly clear is that the modern environment is contributing to the alarming rise in autism. In the 1980s, autism affected 1 in 2,000 children, but today, 1 in every 150 kids will struggle with some form of autism spectrum disorder. Our genetics take hundreds of thousands of years to change, but our environment makes us unhealthier every year. So we can either blame our genetics and watch kids suffer more and more as time goes on, or we can try to make an impact by changing our modern lifestyles. Be politically correct or actually help the kids. The choice is yours. All of these factors we've discussed are environmental factors that directly affect your child after he or she is born. However, there's also many more factors that could cause autism before or during pregnancy of the child. Let us know in the comments if you would like to see a video on these prenatal causes of autism. Don't let them fool you. Subscribe to our channel.